generally where we're headed. You may have just heard Rennie and Beth collaborate there on the best approach crossing this next little bit. We're going to be flattening out and then heading up a steep area on the other side.
Yeah, it looks like Jeff's pulled that up, so I'll zoom out a bit and you can kind of get a better context of the part of the seamount that we're exploring here.
Yeah, thanks, Rennie, for zooming out on our underlying map.
Yeah.
So
it's not even the peak, it just keeps going. This is just a segment of what we mapped today.
Exactly.
Anyone want to take a stab at that coral colony?
Let's take a look.
Oh yeah, there's a nice.
Play a game. Give everyone a week.
Okay, all right.
Is this a bingo game?
Yeah.
Okay.
Rennie's really up for the challenge here.
I'm on it.
Let me get us a better angle for this guy.
It was in the slideshow today.
Is it a lace coral?
Cold.
Cold.
I haven't seen one this dive so far, so it's kind of noteworthy.
I saw that Sarah actually drew pictures of all the things that you. I just wrote a bunch of words.
All right, go ahead on there a little bit there, Jeff. I'll do the jerky. There you go.
Is that a Metallogorgia?
What do you have on your bingo board, Rennie?
Do we need a closer shot? Do we want to?
Yeah, can you go tight on the center of it?
Yeah, let's see. Get you a little bit more centered. Got that spiraling stalk.
We'll see who's home.
My guess is still Metallogorgia.
Oh, it's very lovely.
But I don't think I'm right.
Is that a squat lobster in there? On the top of the frame?
Looks like
yeah.
it.
Something.
Yeah, I think so. Or a shrimp, maybe?
Yeah. Claw.
Did I miss what it was,
Tough to
Steve?
tell. No, I heard somebody say it.
Wasn't me.
Although, something interesting about this colony.
Oh
So
man,
at first I thought it was Metallogorgia.
Yeah.
But
we both thought it was the wrong thing.
But it's actually probably a Chrysogorgia colony. But yeah, Metallogorgia typically doesn't have that kind of morphology to the main axis, the big long branch. Look at the size of that sponge base. I don't know how everybody sees that.
Yeah.
That's got to be 20 centimeters on that.
Let's have a look.
On that pillow. But yeah, I think in the end it was probably a species of Chrysogorgia, but it was convincingly.
Chrysogorgia was my first guess, and then when we got in, I thought it was Metallogorgia, but it seems like you thought the exact opposite.
Yeah, no, it fooled me in the beginning too.
Was the stalk one of the features?
Yeah, the stalk was a giveaway that it wasn't Metallogorgia. So Metallogorgia has more of a straight, dark main stem that goes down to the rock. Whereas the Chrysogorgiids, sometimes they'll be white, sometimes they'll be exposed golden color. But also the branching pattern, branching sequence was very different than Metallogorgia. Yeah, that game didn't go too well. Just because it fooled me. It fooled me as much as it fooled probably all of you.
Is that a sea star there in the... Oh, yeah. Oh, yeah.
Oh, yeah.
Star.
Small brittle star up to the left.
Looks like we'll go over some kind of local peak.
Alrighty.
Looks like we'll go over some sort of local peak that Argus is going over this ridge, kind of.
All right, where are my altered rocks at? We got so lucky right at the beginning of the dive.
Yeah. Tooth extractor.
Do you think that altered rocks is a genre of music underwater?
Altered rock.
It's got to be now.
Hey.
Alternative.
That is my only contribution to humor today. My apologies to everyone. Keep it coming, Jess.
Yeah.
Oh, that's a very cute little sea
We've
star.
obviously got some pun lovers on the...
Looks like a rock star.
Oh, little
I
tiny
think.
star.
Settle down, you two.
So cute.
That is a cutie.
Now there's another sponge base, I think,
To the left.
off to the
there.
left.
Oh, yeah. Oh,
It
the
must
very,
have been
very,
a
very base.
productive area at some point and is no longer with all the dead ones we've been seeing and no live ones or nothing of that type.
That's a good point, yeah.
Big boulder there. This feature is pretty steep. It's almost like a little, in Argus, it almost looks like a pinnacle.
Mm. Yeah, I think we ended up quite a bit downslope on the other side.
So it looks like Jeff has put up the sonar view of Argus, maybe, in the lower left quadrant. Rennie, can you tell us what we're looking at?
Which one is it? That up there?
Yeah.
Yeah.
Okay, so we have two rotary scanning sonars. The one on the left is Argus, and those red rings you see are 20-meter divisions. So from the sensor, which is mounted downward on Argus, when you start to see features, so black is nothing and a bright yellow is a hard return, and blue is somewhere in the middle. So you can see that kind of blob in the upper left quadrant there. That's the feature that we're heading towards. So we get kind of a return off of that. And then those spiral lines you see around the outer edge are interference from another acoustic device we have. And then the one on the right is Hercules. It's mounted on the top facing upward, and those rings are 10-meter divisions away. So you can see that we've got a very stark, bright feature right in front of us, and then when you look at the Hercules HD cam, it's because we're sitting in front of a rock wall. So Argus we like to keep, just because of the unknown heave of the ship and motion, and because we are going at a set speed and can't really stop it as easily at depth. When we stop the ship, Argus tends to still keep going. We like to keep any features around 20 meters out.
Makes sense.
Yeah.
Yeah, so that sonar is definitely, on Hercules, is definitely capturing the steepness of this wall that we are ascending. We've seen some corals and sponges go by. This area definitely, probably a combination of its steepness and current. It doesn't have very much sediment on it at all, even though it's probably many, many, many millions of years old. Here's another, what looks like a coral here. That's something that we've seen before, Steve, on this dive?
Yeah, that's Chrysogorgia we saw recently.
Okay.
At the end of the previous watch. And I can give you a name. Possibly Chrysogorgia flavescens, as what we tentatively ID'd it as. But if the density picks up a little bit, I might be interested in popping another eDNA water sample. So we'll see how it turns out in the next few meters or so.
Yeah, it looks like there's at least a couple of bamboo corals or something like that up ahead.
Yeah, you can really see the nodes on that one at the base.
Sorry, folks. So Steve, would you like that eDNA sample or still hold off for a little while?
Let's see what it looks like on top of this slope.
Roger.
Yeah, it looks like if we go up to the next contour, we might be close to the flat part.
Sometimes you get on the tops of these steeper faces, you'll get a large aggregation of corals along the rim. But the densities are picking up for sure. We got out of that rubble field a while back.
Steve?
Yeah.
The bamboo coral that we've been seeing, the polyps are often kind of this salmon color, pinkish, reddish. Do we know what it is in those animals that gives them that pigment or hue?
No. Yeah, it's tough to say. Sometimes bamboo corals will be completely translucent and the polyps tissue.
looks like
Sometimes
we
there'll
have
be
another.
red or pink.
Yeah.
In very rare instances, some bamboo corals actually have a yellow wash on the base of their colony. So yeah, color is one of those things that folks tend not to pay attention to because it's so variable. But I couldn't tell you why where they get the red color from. I suspect it has something to do with wavelengths, like wavelengths of light being attenuated, but there could be some other component in their tissue chemical reaction that causes that as well.
And earlier today we were discussing whether any of these animals fluoresce, either due to the content of their own body or also maybe from symbionts or epibiont microbes.
Yeah, the bamboo corals, some of them have been shown to have fluorescence. I don't know if it's been examined as widely in these other families like the chrysogorgiids, but sea pens, I know, bamboo corals, to some degree as well. Yeah, okay, I think there's a sufficient density here that we could do a water sample.
Okay.
If that's all right.
Sure thing.
Kind of at this altitude is perfect.
All right. Would this spot be correct for you?
Yeah.
All right, let's do it.
Just as a reminder, we've popped 6 and 4 so far.
So maybe go for 5.
These Niskin bottles that we're about to see are open containers that can be deployed, and when the little trigger is pulled, they're capped, and any water that was free-flowing through that is now captured and won't mix with water as we continue to move through the dive. So that water that was captured contains kind of the sloughed off cells and DNA from many, many organisms living in this particular region, its geographic location. It's really useful for scientists to process and be able to compare the DNA that they find in that water sample to the organisms that they see in this photo series.
Trying to see which color corresponds to 5.
Is it the green one?
Is it green? Okay.
Is it resistor color code?
Yeah, I think it's green.
And go down a little bit. I'm at 6 meters. Go down to 4. All right, Steve, we're at about 4 meters, 5 meters altitude.
Perfect.
Is appropriate for the sample?
Yep.
Great. Pull in now. Oh.
There we go, that's bottle 5.
Yeah, that's correct.
And this is sample number 008.
Roger that.
We just deployed our 3rd Niskin bottle, collecting water samples on this dive and getting reoriented to continue moving.
Yeah, so now that we're on the move, we can see that in the area around where we just took our sample, there's many different coral all around us. So hopefully that'll be a good sample for the UDNA study.
What are those really bright spots that are showing up? Little yellow.
It's a little hard to tell. Moving
coral bases.
Yeah, I'm not sure.
Yeah.
It's hard for me to tell on this screen here. It's not as high resolution as up there.
I was wondering if those might be those little cup coral you were talking about, Steve, or if they're too flat.
It looks like an anemone maybe, or is that a cup coral? Looks like an anemone.
Large anemone there.
Yeah, and as we continue to rise up this ridge feature, we're coming across even more coral of different types.
A crinoid sea star over on the left.
Yep. Oh, and here we actually have a branched.
Yeah, this is what I was talking about before. The thing that drives you nuts about this group is you never know what they're going to do next. Sometimes they branch, sometimes they don't branch. Sometimes they branch halfway up the colony, sometimes they branch at the base, you know. They just do what they want, and they don't care.
Are you saying the bamboo coral is the honey badger of the coral world?
Can we zoom on that branch point just for reference on that bamboo,
Jeff.
just to see what's going on there? If it is a branch or maybe it's some sort of secondary settlement event.
Looks a little thin on the left side of the branch.
All right, go ahead and push it in there a bit.
I'm wondering, since it has kind of this kink in the colony, well, we'll take a look first and see if there's a node at that branch point. If there's a node at that branch point and the branch is emerging from the node, that could be something different that we haven't seen yet that's actually different from these large unbranched colonies we've been observing for most of the dive. Do you have any more zoom on that?
Hey, go ahead, Jeff. I'll hold it steady.
It's tough to tell from here.
Yeah.
It does look like there's some discoloration there. Maybe there's a node, but really tough to tell.
You can see what looks like a node a little further up on the right side as well as on the left. So I'm not sure if it's actually at the node. Or if it's
tough to tell. Yeah, it's the tissue is quite thick and quite a density of polyps too. Tough to tell, but okay, we got some good imagery at least, and
okay,
that'll suffice for this colony. But yeah, this is something we've seen a lot, and we're just not sure if it's a characteristic of something about their growth, or if it's a different species than some of these other ones we've seen. I tend to lean on that there was some sort of aberrant growth form that emerged, or maybe a secondary settlement, like a larvae settled on some bare skeleton from one of the other colonies and started growing and potentially fused with it.
yeah, it did seem a little thinner, like maybe it was a different
or, you know, sometimes if there's a predation event or some sort of grazing, that can cause these growths to appear.
Oh, we got a little fish in the view.
Ooh, maybe it'll be an opportunistic view here. Jeff, you want to do a little partial zoom there?
Not see eyes on that fish. Oh, there the eyes. You're looking at a...
What is that?
Oh, don't go into the vehicle, friend. That is not where you want to go.
Yeah, you definitely don't want to come into the vehicle. That's going to be bad news.
Yeah.
We have any fish specialists in the room?
Is it attacking?
I know, it really
Maybe. He's really adamant on going into the vehicle.
Oh, hello.
Is it
Oh,
is it a
goodness.
snailfish
mode.
or a rat?
Take away the smudge.
It looks like a snailfish. You can see those whiskers kind
Yeah,
of
it does.
tickling down.
Some nice little video clips of fish attack.
Roger.
All right, it seems like we're up on this little flat plateau again. You can see that there's a little more sediment settled on the top of the rocks here. Now that it's a little flatter. All right, and we're almost at the 2400 mark.
Yeah, the 2400, so my rule of thumb is 1800 to 2400 is typically like the sweet spot for a deep coral diversity maximum. So I suspect we're going to probably be seeing more of these, more species appear over the next several hundred meters of distance we travel.
Exciting.
Yeah.
But it's also been quite impressive so far, you know, down to 2,700 meters. It does look a little more turbid up here, a little bit foggier.
Yeah, I'm wondering, so a little over to our east in the direction we're heading right now, it looks a bit steeper. And so maybe we're seeing wash coming up over that. We'll find out when we get there. Jess, how is our ability to stop and pick up anything at this stage?
Oh, that's all good. There's a ship moving, but we can do it fast.
It's okay. We don't need to. I was just curious.
Sure thing.
So if we're talking about that coral bingo, what have we seen so far this dive, Steve? What are the major groups?
Yeah, this it's kind of been opposite a little bit of what I suspected to see down here. Obviously, the bamboo corals I expected to see. I expected to see more than just the one species. I think maybe 2 species of bamboo corals we've seen on this dive. I expected to see more and larger branching colonies. It's overwhelmingly been Chrysogorgia dominated golden corals, and specifically by the genus Chrysogorgia. The black corals were more prominent lower, deeper, and they've since disappeared. I haven't seen very many. And then what else? Primnoids also. We saw a number of primnoid species down deeper, mostly in the genus Narella, and those have also kind of disappeared as we move shallower. Could be a transition, but it's not quite clear enough yet. We have a number of species that have been with us throughout the dive, but yeah, it's not an easy sell to say there's a transition between, X depth and Y.
Rennie, can you zoom out on HYPACK a little bit, please?
Sure.
Thanks. So I'm wondering as we're going here, if we want to maybe change our heading a little bit more south, southeast.
Sure.
To kind of come up that ridge as it's getting a little bit steeper there.
Come up here this way.
Yeah, something like that.
Sure. Yeah, that works. So after we complete this move, I'll go, just up like that.
Trying to convince myself that I'm going to find another rock before the end of our shift.
I think we might just have to sit down and start poking things.
Yeah, I'm looking at these and trying to evaluate if they're just going to be hard or if any of them are going to give. Right now I'm just seeing mostly ground mass. Although that's a beautiful little football right at the top of the screen there. Jess, do you think we have the ability to get a little closer to this one right here?
Sure. Sure thing. I'll be a little at the end of my leash. Actually, no, I'll be good. All good.
Oh, that's big.
Oh, we're not taking that to go, but it is a nice rock.
You can't porch that? Put it in the front porch? Roll it up there and give it a bear hug.
I don't think the next watch will be very happy with this. We can park here for a minute and look at it.
Yeah, no, this is, these all look pretty hard. It's always very deceiving. What you see in the distant field of view always looks, to me, darker than it actually is. Okay, we can keep moving.
Roger. It's very crusty, doesn't it?
Mm.
Yeah, it's almost like a pavement. Sediment dusting.
Oops, sorry.
All right. High packet looks like we're right at a little pinnacle, and that's exactly what this looks like.
Yeah. Wow, it's a cool view.
Pretty
This
cool in
reminds
our
me of like rock walls I've seen around some of the islands.
Yeah.
Yeah.
Looks like we might have hit something with a prop wash.
Let me go back the other way, actually. Take out my wrap. All right, one moment, folks. Just going to take out a little wrap.
Okay.
Thanks, Jake.
So for those at home that might not know what that means, between Hercules and Argus is a tether. We always want to keep track of how many times we've turned around so that we don't put any kinks in that tether. So occasionally need to do some maneuvers, get everything all straightened out.
Oh,
Hercules just did a pirouette underwater.
This side's pretty steep, isn't it?
Yeah.
Getting tugged a little bit.
Raj. I'll come back towards you here.
Did we lose some visibility because the thrusters were turning in all directions, or is this a...
No, we're just off the
current?
wall a little bit.
Yeah, we backed up a little bit while
We're
we
up
did
higher.
that maneuver
Yeah.
so that you don't run the risk of pirouetting into something. It definitely looks like there's a lot of particles in the water here.
All right, from here we're gonna go 175.
175.
175, Raj.
I mean, yeah, look at that marine snow coming down now.
See you on down cam.
Raj.
For our viewers out there, our watch will be ending in about 25 minutes, just over 20 minutes. We're looking for one final rock for Beth, potentially. I want that Oreo cookie crumble crust.
Oh, don't be talking about cookies. I'm hungry.
Those cupcakes are still down there. They won't go away.
I know. There's just
No
I
matter
think
how
he
many
keeps
I
adding. I think he keeps bringing more
keep
out.
adding more.
Yeah.
Fighting an infinite foe.
Makes you wonder what the shelf life of that whipped cream is.
you don't want to wonder.
Yeah, don't.
Don't go
Don't
for the whipped
wonder.
cream ones.
All right, I'm gonna call that in if that's all right.
Sure thing.
Bridge nav. One step 30 meters, bearing 175. currents have definitely changed.
Yeah.
When we first started, Argus was hanging out like 80 meters off there. That was its settled point. Now it's actually close to aft of the ship where it belongs.
Yeah, now it's not, yeah. It has shifted a little bit.
All right, we're on the move again.
Yep.
Still at that roughly 2,400 meter mark. We'll start making our way up a little shallower soon.
How far are we planning on ascending on this dive, Beth?
I don't know. Rennie, that endpoint that we have there on high pack, do you know what the depth is there? I thought it was around 2,200, but I might be wrong.
Yeah, that's right, 2,200.
Okay. That was our original target. That's something we thought we could accomplish in a 12-hour dive. But we are giving ourselves the flexibility that if we're seeing interesting groups of animals, that we could extend the dive and keep going shallower. And if we feel like we've seen what we're going to see, then we can end up calling it. So that'll be the next watch's decision.
Yeah, and the next watch starts at 12 a.m. Hawaii Standard Time. That will be our chance to listen to them or go ahead and go to sleep.
We got something like a 2.5 hour ascent.
Yeah. All right, come on, Rox. Where are you at?
Those shadows can be deceiving, can't they?
And we just put in a new ship's move, right, Rennie?
We did, 175, so we're going a bit more south.
I see something large and white in the Argus view.
Yeah, there's something going to come into view soon here. There it is.
Finally, a live one.
There's a live one of those sponges.
Oh, that's pretty.
Yeah, this one's almost certainly alive.
Much healthier looking than the one we saw earlier.
Mm-hmm. It kind of explains why they all break off right about the same point. They all have what looks like, their thinnest section really, really low on the colony there, and then, they're way up in the flow. They've got this big sail.
Seems like such a narrow stalk to support that mass.
But top heavy.
It's fairly rigid when you feel it, but it's not strong by any means.
And is this an example where it's pretty consistently facing one direction? You're not seeing it throwing off in different offshoots?
Yeah, it's interesting how these sponges tend to, the concavity, the concave section of the sponge is always up current, facing up current, so in this case to the right. I'm guessing to capture and slow the flow. That way, you can actually see it's starting to bend a little bit with the current as well as on top. But corals tend to do the opposite. they tend to like to feed with their polyps down current. And I can't speak for the sponges, but I think the corals like to induce turbulence in the flow. And when you induce turbulence in a flow, it slows down things that allows the tentacles to capture those particles.
These are almost like little broken low bait basalt flows.
Mm. So your gut says if we were to poke any of these rocks right here, they'd be pretty solid.
I am pretty convinced we'd poke them and Herc would just bounce right back off.
Never know until you try.
That's true. Yeah, and I don't know how much more we have for the ship's move, but might make sense to settle in anyway if we're going to have a shift change. But I leave that up to the pilots, obviously, if you think it's safer to back off.
We can call in one more if you want, or we could poke some stuff if you think it's pokey.
Yeah, well, let's poke some stuff.
Okay.
Let's see. Are the lasers on? Yes, they are. That looks good. Definitely not that.
Yeah, I think it's a little hard to say, but maybe 5 or so meters, maybe 10 max. I think we'll be all right.
Okay. So we'll only have a little bit of swing, so yeah, anywhere. What type of rock are you guys looking for now?
I'm trying to find another altered piece. So like we picked up at the very beginning of the dive that had a little squish to it. And I'm just looking at some of these to see if they might be good targets.
Roger. A little bit of squish.
But they all look very solidly attached to one another. I think my eyes are also just getting tired from looking at different shades of gray for 4 hours.
Yeah.
Where?
At 9 o'clock.
Over here?
At 9 o'clock off to the screen there. Off to the left side of the screen.
Over here?
Just kind of a dark, yeah, that rock. I don't know. Does that look?
Yeah, I think there's also like a shadow in that corner.
Shadow.
I keep seeing it too, and then I'm like, wait, no, there's like a oil spot or something on the camera lens.
The tiny little thing bottom
Um,
center,
Tiny little thing.
but I don't know if that's what you're after or if it's attached.
yeah, no, this all is not quite what we need. Maybe something in here, but it's hard. I don't know.
Oh, we got winch feature cam back.
This one here?
It's hard to tell if it's actually coloration or just shadow.
Yeah.
Yeah, so wondering if any of this might be altered.
Jeff, you want to push on in there a bit to center screen?
So I'm wondering if we can try to poke on some of that and see if it's soft or hard.
Yeah, sure thing. All right, full wide there, Jeff, for a moment. Get myself set up proper.
Gonna do a DVL reset.
Roger. Jake, you want to give it a poke?
Poke? Okay. I gotta refresh myself. We're trying to remember the main functions. Okay, so what are we poking here?
I wanted to see if any of this material is soft.
Okay.
All right, Jeff, go ahead and push on in there a little bit. That's good there. Jake, I'm going to pan the camera a little bit.
Okay.
And remember, you can always index.
Open my jaws, you said?
Oh, you can always index and then re-stretch.
Yeah, it seems pretty hard.
Yeah, moving the vehicle a little bit.
Try doing it. Nice. Yeah, it looks looking.
Can you see about these pieces here?
Down there, okay.
I give it a bump.
Yeah, or even that one right there. Either one, any of those. All right, can you try to pick that up and we'll see what it looks like?
Sure. Show this.
Nice. I'm going to change our delta. Nice. Good grab.
All right, can we bring that in a little closer for a zoom?
Stand by there, Jeff.
And I'm not sure what your grip strength is now, but if you could try to give it a squish and see if it has any give to it.
Sure.
If you want, you can move it down a little bit so it's in the camera.
Okay.
We'll just get good light on it before we zoom.
That was full strength.
Yeah, it looks pretty hard.
Yeah.
Yeah, I don't think that's quite what I'm looking for our stuff. It looks just like it's got a thin veneer of alteration, but it's probably mostly ground mass. And it's probably very similar to what we picked up last time for our last sample. I don't remember what number that was, Steve.
So you're returning it to the seafloor?
Yeah, I don't think this is quite what we want.
Okay.
And I'd rather let Andrea pick up the samples that she'd like since it's almost her shift. So thanks, but no thanks.
All right, well, this is
back to its rightful place.
About to conclude our 8 to 12 watch. You'll be hearing some of us muting out as we swap over with our colleagues. Thanks for following along. Stick around for the next watch, and we'll see you at the next 8 to 12.
All right.
Nice. Then shoulder all the way up. Awesome. So do we want to trek around here a little bit more and see if we see some more rocks? Or?
Yeah, if we think that there's time, if we could maybe hop up a little bit and keep having a look around and see if there's anything else that might be soft and squishy.
Roger.
All right. Oh, hello. Whoa, sorry.
Yes.
All right, gonna do a pilot shift over. So thanks, guys.
Thank you.